Artificial intelligence is getting very good at proposing biology. It can help researchers rank disease mechanisms. It can suggest targets. It can explore chemical space. It can help generate candidates that might have been difficult to reach through conventional search alone. That is real progress. But it is not the same thing as proof. This is the first principle for quantum convergence bio. A computational claim earns practical value only when it survives the kind of validation the claim actually requires. If the claim is structural, the evidence burden is structural. If the claim is functional, the evidence burden is functional. If the claim is clinical, the evidence burden is clinical. A model can move a hypothesis to the front of the line. The world still has to test it. That is why the wet lab is the constraint. Not because computation is unimportant, not because artificial intelligence-assisted biology is hype by default. The point is sharper than that. The wet lab is shorthand for the full evidence system: experiments, measurements, controls, replication, trial design, interpretation, and the discipline to say what a result does not yet prove. A useful way to see that distinction is rentozertib. Rentozertib, also identified in trial records as INS018055, is an investigational small molecule TNIK inhibitor being studied for idiopathic pulmonary fibrosis, or IPF. A 2025 Nature Medicine paper describes the program's artificial intelligence-assisted target discovery and molecular design path, followed by a randomized Phase 2A clinical study. That makes it a strong practical case for this episode. It is not just an algorithmic demonstration, it is a chain of decisions that moved from computational proposal into biological and clinical testing: a proposed target, a designed molecule, preclinical work, early human study, randomized Phase 2A evidence, and a registered Phase 3 study. But listen carefully to what that means. It does not mean rentozertib is approved. It does not mean the drug has established safety or efficacy in the broad clinical sense. It does not mean Phase 3 will succeed. It means an artificial intelligence-assisted hypothesis reached a harder evidence layer. That difference is everything. The Phase 2A study enrolled 71 people with IPF in China. Participants were assigned across 3 rentozertib dosing groups and a placebo group for 12 weeks. The study was multicenter, double-blind, randomized, and placebo-controlled. The paper reported that overall treatment-emergent adverse event rates were similar across the study arms. But treatment-related adverse events were more common in the rentozertib groups than in placebo and increased across the tested dose groups. Some participants also discontinued treatment because of adverse events, including liver toxicity or diarrhea. The study also reported a higher mean change in forced vital capacity in the highest dose group than in placebo over the 12-week period. Those findings are meaningful. They are also bounded. The authors note the small size of each study arm. They note the geographic and demographic homogeneity of the participants. They note the short follow-up. They note discontinuations in a study with only 71 participants. So the correct interpretation is not, the artificial intelligence found a proven treatment. The correct interpretation is, an artificial intelligence-assisted program produced an early clinical signal that now has to carry a larger burden of evidence. That is how biomedical trust is earned. The next layer is Phase 3. In July 2026, the company announced that rentozertib had entered Phase 3. The current clinicaltrials.gov record for NCT 076874459 lists the Phase 3 study as not yet recruiting, with an estimated start date of August 30, 2026. It lists estimated enrollment of 320 participants, 47 locations, and an estimated primary completion date in October 2029. The official trial title describes a prospective, randomized, double-blind, placebo-controlled, parallel group study evaluating efficacy and safety over 52 weeks in patients with IPF. The primary outcome is the annual rate of decline in forced vital capacity over 52 weeks. That design matters because it asks a more demanding question. A larger study can test whether an early signal holds with more participants. A longer duration can test whether the signal persists. Randomization and blinding help reduce bias. A placebo control gives the result a comparison point. But a design is not a result. Phase 3 registration is not Phase 3 success. A planned endpoint is not an achieved endpoint. A company announcement is not regulatory approval. This is where public artificial intelligence narratives often run too fast. The story wants to jump from computation to breakthrough. Biology does not let you skip the middle. The middle is measurement. The middle is whether the proposed mechanism behaves as expected, whether the molecule does what the story says it should do, whether an early result can be replicated, extended, and interpreted without turning uncertainty into certainty. Now compare that with a different artificial intelligence biology example, protein structure prediction. AlphaFold family systems changed structural biology because their benchmark performance became strong enough to alter the operating assumptions of the field. AlphaFold 2 especially became a landmark for many single-chain protein structure predictions. AlphaFold 3 is officially framed as predicting biomolecular structures and interactions, and AlphaFold Server is described by Google DeepMind as providing free access to AlphaFold 3 prediction capabilities for non-commercial research. That achievement is real, but it teaches the same lesson. A benchmark can show that a system performs a defined task under defined conditions. It can show that a predicted structure is close to an experimental answer for many targets in a blind assessment. It can make structure-informed work easier to start. It can guide experiments and help scientists form better hypotheses. But a predicted structure is still a model. It does not automatically establish protein function. It does not automatically establish binding behavior. It does not automatically capture dynamics, disorder, post-translational modifications, disease relevance, therapeutic value, or clinical utility. And version distinctions matter. AlphaFold 2, AlphaFold Multimer, and AlphaFold 3 should not be collapsed into one generic claim. Different systems address different benchmark problems with different strengths and limits. So the AlphaFold lesson is not that prediction replaces the lab. It is that better prediction changes what the lab can ask. A predicted fold may help choose a mutation to test. It may help interpret a density map. It may help prioritize a target when no experimental structure exists. But if the claim is function, you still need a functional assay. If the claim is binding, you still need binding or activity evidence. If the claim is drug discovery, you need hit and lead validation. If the claim is clinical, you need human evidence. This is the validation ladder. Computational prediction is one rung. Experimental measurement is another. Functional validation is another. Laboratory utility is another. Translational evidence is another. Clinical evidence is another. The mistake is pretending that success on one rung automatically means success on all the others. Rentosertib is useful because it makes the ladder visible in a practical case. AlphaFold is useful here because it makes the benchmark versus utility distinction visible in a technical case. Together, they show the same rule. Artificial intelligence-assisted biology becomes valuable when the claim and the evidence match. That should change how we talk about the field. The question is not simply, can artificial intelligence generate more biological possibilities? It can. The better question is, can those possibilities be converted into experiments with the right controls, measurements, context, and interpretation? Can they produce results that survive a higher evidentiary burden? Can the field resist turning an impressive computational origin story into a conclusion the evidence has not earned? That is the real bottleneck. The future of artificial intelligence-assisted biology will not be decided only by how many targets, structures, or molecule systems can propose. It will be decided by whether those proposals improve the complete loop from hypothesis to experiment to credible decision. Faster design without trustworthy validation can simply create a larger queue of uncertainty. But faster design paired with disciplined validation could matter. It could help researchers spend scarce experimental capacity on better questions. It could make the handoff from computation to laboratory work more deliberate. It could make the evidence layer more important, not less. That is the frame for Quantum Convergence Bio. We will cover the models, the molecules, and the platforms. But the channel will keep asking one evidence. First question, what kind of validation would this claim require, and has that validation actually happened? If you want evidence, first artificial intelligence biology coverage, subscribe to Quantum Convergence and follow the Bio series as it develops. Because in biology, prediction is only the opening move. Trust is earned after the test.